I'd like to introduce our next guest, Dr. Robert Nagourney. He's going to speak on cancer biology as systems biology, implications for developmental therapeutics in oncology. Dr. Nagourney is Medical and Laboratory Director at Rational Therapeutics in Long Beach, California. He's board certified in internal medicine, medical oncology, and hematology. Let's see. He has authored more than 100 manuscripts, book chapters, and abstracts, including publications in the Journal of Clinical Oncology, Gynecological Oncology, and the Journal of the National Cancer Institute. So, Dr.
Good afternoon. Thank you for staying late to the afternoon to hear this presentation. I'm very delighted to be here, actually. I am extremely impressed by the lectures that I've heard. I didn't know, I've never attended this conference before. I come from a general allopathic medical background. I teach pharmacology at the University of California, Irvine. And I've been engaged in laboratory analysis of human cancer for many years. And I have, for many years, had an interest in botanicals and natural products and a more global approach to human cancer. And it's incredibly impressive to me that this group are so sophisticated in your understanding of molecular pathways, signal transduction, and many of the phenomena that are so much front and center today at the American Association of Cancer Research, which I just returned from a couple of weeks ago. In point of fact, you'd be surprised how similar your presentations are. So I'm hoping, there's a glimmer of hope here, that we'll actually get these two camps together and do something very productive, because I think there's a great deal of potential here. And I'd love to think that we don't silo all these resources. It's of interest. I was listening to the presentation just before me on drug resistance reversal, and I was thinking, people sometimes say that I look like Patrick Stewart and different actors, Ben Kingsley sometimes. But I think that the last fellow looked like Oliver Reed. At least that was my take on him. In any case, it was of interest to hear his presentation because I actually began my field of formal investigation in the reversal of drug resistance. That was where I first started working in the area of cancer. And so we had developed a pharmacologic manipulation of drug resistance mechanisms to poison the drug resistance efflux pumps, the MDR. And we thought we would use all of the available chemicals that were available to us at the time. So we put together a combination we called VILCH, which was verapamil, imipramine, tamoxifen, chlorpromazine, haloperidol, with or without menadione. And we used to add that into tissue cultures, and we actually could reverse drug resistance, and we developed clinical trials and treated patients with infusions of therapies and oral tamoxifen, and it actually worked. Unfortunately, it didn't work for very long. And the reason it didn't work for very long is because cancer cells are very clever, and they evolve continually. And as you block one pathway or one mechanism of resistance, they will develop another and another again. And so drug resistance reversal must be much more global than just pathway mediated. We have to be broader in our thinking. So what I'd like to talk about today, and what I'll spend the next few minutes talking on, is the concept of human tumor biology in a tissue culture environment, a window on cancer biology. So tumor biology as a window on human biology is a very interesting opportunity for us to learn about all diseases. Cancer is a perturbation in normal physiology, but it is deeply connected to heart disease and diabetes and infection and degenerative diseases and dementia. These are all parts of one long continuum. As you know better than most, disease entities are more closely aligned. And so we can look at cancer as a window. It gives us insights, as does AIDS and other areas of disease, so that we can begin to look at normal physiology differently and perhaps different disease states using drugs that might not originally have come to mind as therapies for those purposes. I was speaking with Dr. Boyd a few minutes earlier, and we were talking about drugs that are used as antibiotics that are downregulators of NF-kappa B. So that there are all of these cross connections that are so open to our exploitation if we would just keep our eyes open and watch for this. So if we think of cancer biology as a window on human tumor biology, then we go back to the 19th century when Rudolf Virchow, one of the founders of modern medicine, said omne cellula e cellula. And what he meant was that every cell originates from another cell existing just like it. They all come from each other. Now, the problem was that led to our thinking that cancer represented abnormal cell division. If cells divided to create tissues and organs and bodies, then abnormalities must be abnormalities of cell division, and we became extremely focused on the process of cell division and mitosis and DNA synthesis. This led us to develop therapeutics that were largely destined to attack DNA utilization, the mitotic spindle, and other mechanisms. We can see here about a century's worth of therapeutic development going all the way back to the origins of radiation, the ultimate DNA damaging approach, to the various radiomimetic chemotherapeutics, and finally some of the newer chemotherapeutics, all pretty well destined to do one thing well: stop cells from dividing. Well, radiation, as I mentioned, cytotoxic chemotherapy, combination chemotherapy, the reversal of drug resistance, which we just heard about, dose intensity and dose density, and finally bone marrow transplantation, the ultimate extension of dose density and dose intensity, giving people drug doses so severe and so toxic that they actually kill the patient and they have to be rescued with their own bone marrow or that of someone else. Well, the result? Well, rather uninspiring. This is an example that anyone who goes to a cancer meeting will know only too well. The survival curves that look like railroad tracks going to the ground. No distinction from treatment arm to standard treatment arm. Very little change in outcome, yet many of these trials are deemed positive and are reported and lead to FDA approval of drugs. Marginal, clinically insignificant, yet statistically significant improvements that have powered many of the developments that we use today in cancer therapeutics. Why? Why has it been that cancer therapies developed as we've done so? Why has it been that cancer therapies targeting cancer cells as proliferative entities, why has it been that this has failed to give us the information and the results and the clinical results that we so desperately need in medical oncology? I like this diagram. Rabbit or duck? It's the classic picture of a paradigm shift. Look at it one way, it's a rabbit. Look at it another way, it's a duck. Many things in science are driven by these sorts of phenomena, where you look at it one way and you're sure it's one thing, and then you look at it another way and you're sure it's something else, and sometimes you can't go back from one to the other. Once you've deemed something driven by one process, it's very difficult for you to go back and remember how you thought of it. And once we went into the track that cancer was a disease of cell proliferation and an abnormality of cell division, it was very difficult for anyone to go back and examine any other possible pathways. So you come to the concept of paradigm shifts, as Thomas Kuhn in 1962 described, the concept of scientific thinking, where an accepted phenomenon is suddenly explained by a new possibility, by a new explanation. You look at the sun going overhead, or you look at the horizon, you're convinced the earth is flat, it isn't. You're convinced that the sun is going around you, it isn't. And all at once, you never want to go back and think the same way about what you were sure you knew just a day before. So in a way, we have to change the paradigm of thinking, the concept of cancer biology, so that we have a better concept and a more productive model for the development of therapy. The discovery of programmed cell death, for me, was a more important paper in 1972 than the 1953 description of the double helix. The double helix has provided us a tactic in cancer biology. The concept of programmed cell death has provided us an underlying strategy for all cancer therapy. Recognizing that cancer represents a disorder of cell survival, not proliferation, recognizing that the fundamental process of cancer was different from what all of the people in the last century thought, enables us for the first time to tackle this problem effectively. It turns out that cancer indeed is driven by survival signals, and those survival signals are so critical and so long held that the Xanarabtus elegans, a roundworm of 1,031 cells, is the perfect model for the same process that drives cancer. It turns out that the genes, the CED genes, SED9, which is the anti-apoptotic BCL2, BCLXL, and SED3, the pro-apoptotic BAX-BAC, those genes are largely conserved in our own bodies hundreds of millions of years later. The same genes can be probed. We have basically Xanarabtus elegans genes in our own cells, and those are the BCL2 family and the BAX-BAC family, the anti- and pro-apoptotic proteins. What's interesting is that it's the interplay between these genetic elements that drives cancer, not a proliferative signal, not a capacity to divide, but a capacity to stay alive and live long enough to divide more times than your neighbor. So cancer is driven not by its capacity to replicate, but by its ability to remain alive long enough to replicate at the normal rate. We thought for the many years that cancer was being treated by damaging the potential, the capacity of cells to divide. So we came up with clever ways to damage DNA and disrupt mitosis. Radiation was the first developed in the last century. Thereafter, radiomimetic chemotherapeutics. Thereafter, antimetabolites. And after that, antimitotic substances, the taxanes, etc. All of these seem to be causing cells to disappear in patients because we would stop the cells in their tracks from dividing. But it isn't at all the case. No drug that works in cancer today works because it stops cells from dividing. The taxanes, we think, oh, the mitotic spindle is inhibited and the cell cannot pull its DNA chromosomal components apart, and therefore the cell will be inhibited in proliferation. No, not at all. When the taxanes induce an injury in a G2M arrest, BCL2 is phosphorylated and the cell loses a survival signal. When the antimetabolites are incorporated into the cell reads the antimetabolite as an abnormality that forces the cell to take inventory, and the cell kills itself. Chemotherapy doesn't murder cells, it convinces them to commit suicide, and the suicide is one of many programs of cell death, apoptosis being one. Interestingly, there are many more cell death genes than cell life genes. In fact, from the outside in, death is the norm and life the aberrancy. We are programmed to kill every cell in our body as well as we can, and it is only that rare cell that has the capacity to stay alive despite that. So BCL2 family members numerically, demonstrably outnumber the anti-apoptotic proteins. The anti-apoptotic proteins are the most dangerous. Any cell that stays alive in your body can live to become malignant. So the body is desperately trying to eradicate cells in the body, and only those cells that are given the most survival signals, the stem cells, the proliferative capacity of the reproductive organ, those that line the inside of the gut, only those cells that are absolutely must stay alive are given enough of that essential life force to stay alive against all efforts on the part of the body to continually eliminate cells. Cancer does not grow too much, it dies too little. As I became increasingly convinced of this work in the last years, I realized there was a very good reason why no one had ever really come up with a good laboratory model for cancer testing. All of the tests were being run in an environment where the cells were propagated, promoted to grow, and then we measured proliferation using thymidine incorporation with tritiated thymidine, looking at colony formation, looking at growth to confluence. If you read the literature, you'll find that virtually every laboratory model that was developed was growing cells, adding some agent into the mix, and trying to inhibit growth. So we had a proliferation-driven scientific field, and it turns out that proliferation in cancer is virtually irrelevant. Every cell in your body is growing at some rate, from the hair follicle, which I don't have a great number of, but to the bone marrow, to the inside of your gut. All of these cells are proliferating rapidly. No wonder the drugs that we use have so much toxicity. the bone marrow suppression, the immunosuppression, the loss of hair, the gastrointestinal side effects, these drugs work perfectly well. They're not bad drugs. They're just badly designed drugs. They do the wrong thing. So in the laboratory, we wanted to come up with models that would accurately reproduce what was occurring in patients when chemotherapeutics were administered. And those models needed to be driven by cell death. So we needed to come up with a relevant laboratory endpoint, correlate the findings, and then go forward with the evidence that we could improve therapeutics. These are efforts over the years of different investigators who tried to do this sort of work. And interestingly, the first effort of all in 1954 by Black and Spear was actually a cell death measurement. Black and Spear used tetrazolium reduction, what today would be an XTT or MTT assay, to see mitochondrial function inhibition. That's a cell death metabolic assay. The problem was in 1954 there were all of 5 drugs available. And even though the assay could pick amongst the drugs, it didn't matter. You could actually give every patient aminopterin and uracil mustard and see if they worked. It wasn't until decades later, when all of the synthetic chemists had come up with all these new drugs, that anyone went back to revisit this. And by then, the concept of cancer as a proliferative abnormality had become so entrenched that no one went back and rethought what Black and Spear had done. And it isn't until the last few years that people have come back to the concept of cell death and survival signals as fundamental to cancer. So the dichotomy between cell death and cell growth isn't just a different way to do it, it's an absolutely fundamentally night and day different way to do it. You cannot use growth-based studies to learn about cell death. So this is a picture of Iguazu Falls, the Argentinian side and the Brazilian side. I'm not sure who's the death and who's the growth. I think that would be an argument between the Argentinians and the Brazilians. But it is an indication of the divide between these phenomena. So in the laboratory model that we have been developing, the first paradigm, the fundamental difference from what had been done in the past, was to measure that which we considered to be crucial to accurate prediction